Hi there, and welcome to this run-through of the AQA A-Level Biology Paper 3 from 2018. So this paper 3, is typically considered the most challenging of the 3 A-Level AQA papers. It has quite a strong focus on AO3, that's evaluate type stuff, and there's of course the essay. And the reason I'm starting with this 2018 paper, if you're sitting this in 2024, I would say probably the best papers for you to be doing are 2018, 19, 20, and then maybe 21 as well, because those papers are more likely to have content that is similar to what might come up in 2024. Okay, so let's get into it. Okay, question 1. Broken bones are repaired by cells called osteocalts and osteoblasts. Osteoblasts secrete a hormone called osteocalcin in an inactive form. Okay, that's interesting. Osteocalcin is a protein. The active form of osteocalcin binds to a receptor on beta cells in the pancreas, stimulating them to release insulin. Osteoblasts have receptors for insulin. Osteoblasts have receptors for insulin, okay. Figure 1 shows the production of osteocalcin by osteoblasts is controlled by positive feedback. So the beta cell releases insulin that stimulates the osteoblast to release osteocalcin, which is in an inactive form, and then this osteoclast secretes the acid, changes shape, makes it become active, and then that stimulates the B cell to produce more insulin. Okay, got you. All right, what's the question? Secretion of osteocalcin by osteoblasts is controlled by positive feedback. Use information from figure 1 to explain why this is positive feedback. Okay, so positive feedback is where an initial release of something or initial change causes more of a change, but I think that's a little bit general, so we want to kind of maybe describe the feedback loop. So we might say that a beta cell, when it releases insulin, this stimulates the release of osteocalcin, and then osteocalcin stimulates the beta cell to release more insulin. So therefore, I'd add this, so therefore any initial release of osteocalcin triggers more release of osteocalcin in a feedback loop. Okay, let's check the mark scheme to see if that's kind of along the right lines. Yeah, osteocalcin causes more release of insulin, and insulin causes more osteocalcin release. So, right, let's move on. The secretion of osteocalcin in an inactive form by osteoblasts is controlled by, oh no, we did that one, so my bad. The acidic pH conditions created by osteocalcin cause the inactive form of the protein osteocalcin to change. So suggest how. Suggest how. So this is going to be some form of change to the tertiary structure. So pH conditions might affect hydrogen bonds. They might affect ionic bonds that hold the tertiary structure together, causing a conformational change in the protein's tertiary structure, changing shape so that the active form can then bind to receptors on the B cell. Does it say receptors? Yeah, osteoblasts have receptors on the B cell. Yeah, beta cell. Okay, let's check to see if that's about right. Yep. So pH changes or breaks ionic or hydrogen bonds, either one, and that changes the tertiary structure such that it can bind to the receptors. (1.3) Binding of insulin leads to increase in the rate of respiration in cells such as osteoblasts. So for this, this is more a knowledge thing, so we need to know what insulin does. So insulin, when it binds to insulin receptors, it causes glucose transport proteins to go into the membrane of any body cell, including osteoblast. So this increases the permeability of the membrane to glucose, so more glucose is taken in via facilitated diffusion, providing more glucose for respiration, increasing the rate of respiration. Yeah, okay, that's it. More transport proteins or carrier proteins for glucose. The actual name of it is well, either the GLUT1 or the GLUT4 protein. You might have remembered that. It has come up in previous exam questions, so perhaps you remembered that. And therefore more glucose enters the cell. Question 2. Chronic lymphocytic leukemia, CLL, is a cancer that affects some B cells of a person's immune system. Okay, these are B cells, not beta cells. It's slightly confusing. The first question is about beta cells. B cells produce antibodies. We need to remember that. So I would encourage you, especially when questions get difficult, to kind of annotate as you go along. Lymphocyte, okay, so obviously lymphocytic leukemia. A B cell is a type of lymphocyte, a B lymphocyte. Rituximab is a drug used to treat CLL. Now, if any drug, by the way, any cancer drug ends in MAB, that stands for monoclonal antibody. So I already kind of know I already kind of know how this works, maybe a bit. And if you're thinking about being a doctor or something like that, then you should know that as well. If enough rituximab binds to a B cell, okay, it binds to a protein called CD20, I like this, on the surface of B cells. If enough rituximab binds to a B cell, it can kill the cell. Rituximab kills both healthy and cancerous B cells. The body then produces new B cells, okay, from stem cells. So I might already sort of be thinking about how stem cells. Okay, the amount of CD20 on the surface of B cell varies from one person to another. Okay, doctors investigated the relationship between the amount of CD20 on the B cells of a patient and the percentage destroyed. Okay, so this is results that we would sort of predict. The more CD20, the greater percentage of B cells. There is some variation, but as a sort of general trend, it's like that, isn't it, roughly? Each cross is the result for one patient. Okay, so we're not looking at enormous quantities of data. We can actually count it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. There's just 15 patients here. What statistical test could the scientists have used to determine whether there was a significant relationship between the amount of CD20 on the surface of B cells and the percentage of B cells destroyed by rituximab? Okay, so this is the correlation Spearman's rank. It's called Spearman's rank correlation coefficient. And the reason is we're looking to see whether there is a statistical link between two variables that may be related. Okay, on to 2.2. From the data, what can you conclude about the effectiveness of rituximab in treating patients with CLL? Do not include considerations of statistical analyses in your answer. Okay, so we go back up here and we look at this graph. What can we conclude from this graph? We need 3 points. Well, I think the first point that we can see here is that the percentage of B cells destroyed by rituximab increases the more CD20 there are CD20 receptors there are on the surface of B cells. So therefore, this medication may be more effective if patients have more CD20 on the surface of their B cells. So basically describing this correlation. Another point, it never reaches 100%. So we never destroy all of the B cells, even if we're this highest, the patient here with the most CD20 on the surface of the B cells, like approaching 16, whatever unit that is, they are still having under 80% of B cells destroyed. Now that would mean that potentially some B cells would not be destroyed, and they could go on to divide again to become cancerous again. I presume this cancer, this CLL, is kind of B cells dividing and becoming too high in number. That is what leukemia is. So that's 2 points. What can we conclude? I'm looking for a third point. I mean, we've only got 15 people here in this whole study, so maybe that's worth saying. It's difficult to say for sure, as there's only been 15 people, 15 patients. That's what I'm going to go for. There's 3 things there. I wonder if it's going to get the marks. Let's have a look. Okay, so the first mark is for describing the correlation. The more CD20, the more B cells are destroyed. The second mark is for saying that at best it only destroys about 80% of the B cells. So therefore, I think this is this is also what I said. It won't cure the cancer because some of these B cells could divide again to come back to be cancerous again. This was something obvious that I could have missed. Included little effect below about 5 CD20 on the cells. So let's look at that on the graph. Yeah, okay. So I mean, if we look at this graph, maybe this correlation that I drew earlier is a bit simplistic. So what could we sort of say? Maybe if we were doing more of a line of best fit approach, it would kind of be more like that. So that means that there's a little effect below this number here. So this bit, not much effect, and then more of an effect. And I think that's probably a key takeaway for when looking at trends or scatter plots. This is a scatter plot. See if you can break down the graph into a more detailed trend, not just increases or decreases, but at what point does the gradient get steeper or flatter and that sort of thing. Okay, let's keep going. All right, on to this next part of the question. Approximately 11% of people with CLL also have mutations of a gene called Notch1. This leads to production of a non-functional transcription factor associated with CD20 production. The doctors determine the median percentage of B cells destroyed by rituximab in people with CLL who had Notch and those who did not. Okay, so if people had this Notch1 mutation, it's a pretty low percentage of B cells destroyed, and if they did not have the Notch mutation, then it's much higher. Okay. Human blood contains approximately 1 billion, 1.9 times 10 to the line, B cells per litre. Use the median values in Table 1 to calculate the difference between the number of B cells per dm cubed in the blood of people treated with rituximab with the Notch1 mutation. Use the median values in Table 1 to calculate the difference between the number of B cells per dm cubed in the blood of people treated with rituximab. Okay, so people have 1 billion. All right, so people generally have 1 billion B cells per dm cubed, and we're talking about people who have taken rituximab. So if people who have taken rituximab and have this mutation, that's 1 billion minus 4%, and then people who haven't had rituximab, people who do not have the Notch1 mutation, it's 1 billion minus 22%. So the difference is going to be the difference in percentages times 1 billion. So 22% minus 4% is 18% difference. Okay, so what we're looking for is 18% times 1 billion. So it's 1.0 times 10 to the 9 times, this is 18%, so I'm looking for 0.18 times that, and that's going to give me my number, and that is going to be 180,000. Yeah, that's going to be 180,000. 180 million. I just got that wrong. 180 million. Oh, look, it says expresses my answer in standard form. So that is going to be 1.8 times 10 to the 1, 2, 3, 4, 5, 6, 7, 8 times 10 to the 8. Yeah, cells per dm cubed. All right, let's check it out. See if that's right. I hope it is. 1.8 times 10 to the 8, that is correct. 1.8 times 10 to the 8. Okay. Use all of the information so that's all the graphs, to suggest how the mutation of Notch1 led to the difference in the percentage of B cells destroyed by rituximab. Okay, so this is where we need to like link information in different places. Okay, so what do we know? We know that Notch1 is a transcription factor. Where is that? Non-functional transcription factor. So I'm guessing Notch1, if it produces a non-functional transcription factor associated with CD20, this would probably mean less CD20, I think, and that fits with this observation here that it's only 4% destroyed, whereas if they do have this, do not have the Notch1 mutation, we have more CD20. Okay, and that links to this graph up here. So that is using all the information, because we're thinking that people who have the Notch mutation are kind of down here, something, I don't know, maybe in this region. Oh, and we're looking for, what is the percentage again? The percentage is 4%. So that is going to be, you know, around here somewhere. This is like 10, so 4, yeah, it's going to be in this area here, 4%. People with Notch mutations can have very little CD20, and the median for people who don't have the Notch mutation is 22%, so that's like up here somewhere. So this is no Notch. So how do we put this in an answer for 3 marks? So we would suggest that the Notch1 mutation leads to a non-functioning transcription factor, which means that the mRNA for the CD20 receptor on the surface isn't transcribed, because remember, transcription factors cause RNA polymerase to bind, cause the transcription of a gene. So we could say that the Notch1 mutation leads to a non-functioning transcription factor. This means that RNA polymerase cannot bind to the CD20 promoter, and therefore less RNA for CD20 is produced. So therefore, we're producing less CD20 on the surface of the cells, and then rituximab cannot bind to that receptor, because that's what it does, isn't it? I think it is. Yeah, it binds to the CD20 receptor, and therefore if it's not binding to the cells, then it's not going to kill the cells. That should do it. Let's check the mark scheme. Okay, so what I've got 3 marks. When I was talking about it, I said transcription factor cannot bind to the CD20 gene promoter, I said. So therefore no mRNA or less mRNA is produced, and therefore little for rituximab to bind to, or rituximab cannot bind to the CD20, so no B cells destroyed. So that would have just about got it. This was also worth saying. I should have said this. You can always just sort of have a guess as to what the mutation is. So we need to know that mutation often changes the tertiary structure of proteins. So the mutation changes the structure of the transcription factor, can't bind, no mRNA produced, therefore no CD20 on the surface of the B cells, and therefore no killing of the B cells. Okay, let's keep going. Question 3. This one looks to be about Down syndrome, possibly. Let's have a look. In women, the first division of meiosis produces one daughter cell that has almost all of the cytoplasm. The other daughter cell consists of a nucleus surrounded by a very small amount of cytoplasm and a membrane. The very small daughter cell is called a polar body. Here it is. Polar bodies do not usually divide, and then the same thing happens in the second division of meiosis, resulting in 1 egg and then 2 polar bodies. And the diagram shows this. It also shows what happens to one pair, I'm going to highlight this, one pair of homologous chromosomes. This pair carries 2 alleles of gene A. Okay. All right, it looks like we're going to have to fill this in. All right, so we've got 2 big A's and 2 little A's. So we've got 2 big A's, 2 little A's. Okay, well, looks already, I can see that there's been some crossing over here, because we've got the little ones here, the recessive and the big, are on each chromosome, but now we've got a big A little a there, so we must have a big A little a here. There's been crossing over. Right, this is the one that divides, and that's got to be, if we're basically just counting things up. Look, there's 2 big A's, 2 little a's, and now we've got a big A, a little a, so that's got to be little a there. Okay, so we've already done one of the questions. Done that. Whoops. Okay, cool. Done that. 3.2. Put a tick in the box next to the name of the process that produced the combination of alleles. We've already talked about this, so this is crossing over. So crossing over is where we have an exchange of alleles between non-sister chromatids, between so these two. This is a definition that people always get wrong, by the way, so don't feel worried if you're struggling with this. These two, I'm going to colour them in green, those 2 chromatids on the same chromosome. Those are called sister chromatids on that chromosome, and they have the same alleles. I'll colour these ones in blue over here. These 2 chromatids on this chromosome, those are also sister chromatids, but if we're looking at an exchange between a green and a blue, that's a crossing over, an exchange of alleles between non-sister chromatids. They're homologous, which means they're the same size chromosomes with the same gene loci, but they may have different alleles. Okay, let's keep going. A scientist measured the diameter of a polar body and the diameter of the nucleus inside of it. Okay, so we've got polar body, a little circle, and then a diameter of the nucleus inside it, a circle within inside the circle, a sphere within inside the sphere. The diameter of the polar body was 10.4 micrometers, and the diameter of the nucleus was 7 micrometers. Ooh, the density of the mitochondria in the cytoplasm of the polar body was that much per micrometers cubed. Calculate the number of mitochondria in the polar body. It might help to kind of draw this. We've got a sphere, imagine this is 3D, and we've got a nucleus inside of it, and then we're trying to work out how many mitochondria are in this kind of space of cytoplasm that remains. So first, what we've got to do is we've got to work out the volume of the whole thing, subtract the volume of the nucleus to figure out how much cytoplasm there is, and then we can use that volume of cytoplasm times this density to work out the number. Now the first thing that you need to spot, you might miss it, is that these are diameters. So we're going to want to put these into radiuses. So this is going to be 5.2 is the radius, and this is going to be 3.5. Okay, so it gives you the formula of the sphere. It doesn't always do that. You should know that, 4/3 pi r cubed. So we're going to do, 4/3 pi times 5.2 cubed, that, minus 4/3 pi times 3.5 cubed. That's going to give me 589.0 minus 179.6. So that's 409.4. And that is micrometers cubed. Now that is the volume of this yellow area, and then we're looking at the number of mitochondria, so I then have to times it by the number of mitochondria, the density of mitochondria, so times it by 0.08 times 0.08, and now that gives me 32.75, so 33 mitochondria. Let's see if this is right. Yeah, 33. So I got 32.75. So yeah, 33 is correct. Phew. Okay, 3.4. Mitochondrial diseases are caused by faulty mitochondria. All of a person's mitochondria are inherited from their mother via the egg cell. This is probably something you should know anyway. An egg cell contains approximately 3 × 10⁵ mitochondria. Okay, a proposed treatment to prevent passing on faulty mitochondria involves removing the nucleus from an egg cell donated by a woman with healthy mitochondria, replacing this nucleus with the contents of the polar body from a woman whose egg cells are affected by mitochondrial disease. Okay, how does this treatment prevent inheritance of mitochondrial disease? Okay, well, the first thing you need to know, the egg cell donated by a woman with healthy mitochondria contains mitochondria, but then if we remove the nucleus, we're removing all the genes, all the chromosomes from that woman who has donated the egg cell. However, there are just a few genes on the in the mitochondria themselves. I think it's about 13 actually, not very many, in the mitochondrial DNA. So the mitochondrial DNA will be good, but all the other DNA is going to come from the polar body. So the polar body, just to go back up here, I presume we would want to use the second polar body is the one we would use, because the second polar body is haploid. And that's what we want the nucleus that we use to be. We wouldn't want to use the first polar body because it's a diploid. Well, it's not diploid, sorry, but it has chromosomes that are already replicated. So we wouldn't want to use this one, we'd use the second polar body. And then that would provide half the genetic information, and then we'd presumably fertilize it with a sperm cell in IVF to provide the rest of the genetic information. So, to summarize, we remove the genes from the healthy egg. We have the mitochondrial DNA that is non-mutated, that will provide healthy mitochondria, and then we're going to use the polar body to provide the genetic information from the mother. Let's check. Okay, so the egg has the nucleus DNA genes of the woman. This is the egg created. It has mostly many lots of normal mitochondria. Yeah, many lots of normal mitochondria. There are few faulty mitochondria. Yeah, so you need to be careful that when you're this mark scheme isn't very well described. It's kind of difficult to interpret here. But you need to be careful about where the nucleus is coming from. The nucleus is coming from the woman who made the polar body, not the donor. That's the key thing. Let's keep going. Okay, if most of the mitochondria in the cell are faulty, this prevents many important enzyme-catalyzed reactions from taking place or slows them down. Suggest and explain one reason why. 2 marks, should be fairly simple. So, first of all, I think we need to say that the mitochondria is the site of aerobic respiration and produces ATP. And then we need to say that ATP can be used to provide a source of energy for many metabolic reactions. And therefore, if we don't have enough ATP, we would not be able to drive these enzyme-catalyzed reactions forwards, I think. Let's see if that is what we need to say. Yeah, so not enough ATP produced, and ATP provides energy for reactions. So this is something that maybe is not hugely emphasized in textbooks or in the spec, but sometimes, well, if reactions release energy, are exothermic, they will proceed sort of automatically. A plus B produces C plus D automatically without any input of energy. But sometimes we have chemical reactions that are, in a sense, endothermic. They need an input of energy, and ATP can provide that energy. It can push reactions forwards, providing a source of energy. Okay, so that's the type of reaction that we're referring to here. Okay, now we go on, and we're now on 4. Did I miss a bit? No. Okay, binding of one molecule of oxygen to hemoglobin makes it easier for a second oxygen molecule to bind. Okay, I've dropped in a little picture to illustrate a little bit here. So this is an American spelling with an H-E. We like this A-E. So hemoglobin, first of all, is a protein with a quaternary structure. It has 4 polypeptide subunits. Now those subunits are held together quite tightly. So when the first oxygen binds, it's basically a little bit difficult to get into any one of the subunits and bind to the heme molecule. But when the first oxygen binds, it causes the whole protein, the quaternary structure, the shape changes slightly, making it more what we call a more relaxed form. And this form, it opens up slightly so that it uncovers or reveals oxygen binding sites so that they're more easily accessible. This whole thing is called positive cooperativity. So that's what you need to say. So when the first oxygen binds, it causes a conformational change in shape of the quaternary structure such that heme molecules within hemoglobin are more easily accessible for other oxygens to bind. And this is called positive cooperativity. Right. Let's check the mark scheme, make sure that's exactly what we need to say. Ooh, why have I gone all the way down here? Okay, yeah, so binding of the first oxygen changes, you could say, the tertiary or the quaternary structure of hemoglobin, uncovers another binding site or uncovers another heme group. We don't get any marks just for saying positive cooperativity without explaining it. We need to explain it like this. Okay, let's keep going. Ooh, a hemocytometer. Haven't seen this in AQA. A hemocytometer is a special microscope slide which can be used to count the number of blood cells in a sample of blood. This is on the OCR spec, so perhaps if this is something you want to look at and think about more, you could find some questions from an OCR paper that have this on. Okay, anyway. The surface of the slide has many small equal-sized squares marked on it. The depth of the liquid under each square is 0.1 millimeters. When counting cells that touch the top or left lines are counted, but cells that touch the right or bottom lines are not counted. Doctor used a hemocytometer to determine the number of red blood cells per millimeters cubed in a blood sample. He diluted the original blood sample by a factor of 200 times before putting on a hemocytometer. This is just mean, I find. This is like, so this isn't on the AQA spec. You won't find it in an AQA textbook. You will find it in OCR, but they're basically getting you to do an OCR question, just not having taught you about it. So that's nice. Let's give it a go anyway. I presume they're going to get us to calculate. The doctor counted the red blood cells in many small squares. The mean number of red blood cells per small square was 7. Okay, so 7 is the mean, but we need to work out the volume of the small square. So the volume of the small square is something we're going to need to work out. Right, so how do we work out the volume? Well, the volume, we need to know the depth, the width, and the length, right? So the depth is this, 0.1 millimeters, and then here is the width and the length. Okay, so the volume is going to equal 0.1 times 0.05 times 0.05 millimeters cubed. Let's just do that real quick. 0.1 times 0.05 times 0.05 equals, that's a very small number, 0.00025 millimeters cubed. Okay, so that is 0.00025 millimeters cubed, and it was 7 in that volume. So 7 in 0.00025 millimeters cubed. So then we basically divide by this to work out what it would be in 1 millimeter cubed. So 7 divided by that number. So let's do that. 7 divided by 0.00025 equals 28,000 per millimeter cubed. But it also gives us this information. The original blood sample was diluted by a factor of 200 times. There was something up above about dilution, so let me just quickly check that again. Okay, they just repeated it. Okay, that was nice of them, just so you didn't have to remember it from up here. He diluted the original blood sample by a factor of 200 times. It's written down here again, 200 times. So that means that we're going to need to times this number, times it by 200 to get the original answer, and it's going to be a big number. Wow, look at that. That's amazing. There are 5.6 million red blood cells in every millimeter cubed of your blood. Amazing. Does it say you have to do standard form? Oh. 1 mark for me, not 2 marks. Gotta check this out. Give your answer in standard form. And I forgot to do that. So yeah, 5.6 times 10 to the 5 is the correct answer. 5.6 times 10 to the 5. And if, like me, you didn't read that little bit of the text and you just wrote this, you would have got 1 mark. When counting, cells that touch top or left lines are counted, but cells that touch right or bottom lines are not counted. What are the reasons for this? Well, the reasons for this is to avoid double counting, but I'm trying to think how you would get 2 marks. Because imagine if you were also looking at a square over here. Like, how do you know which, where to count a certain cell? And it is touch the top or left lines are counted. So that means that this one would be counted in this one, and this one would be counted in this one. But this one would not be counted in this square, but if you were counting a square to the right, it would be counted in that square. So it's to avoid double counting, and it's a standardization of the procedure, is what I would say. I haven't seen this type of question before, so we will have to check. Okay, So to avoid double counting, that is the answer that I think we should definitely remember. I wonder if I said to have a standardized procedure, it might be given to be consistent, to have a consistency. Yeah, so. And to avoid dealing with parts of cells. So for example, you don't want to give a number that is, for this one, 1, 2, 3, 4, 5, 6, 7, 7.5 cells or whatever. You don't want to be dealing with half cells. Okay, let's keep going. The doctor also wanted to know how many white blood cells per millimeter cubed there were in a different sample of blood. To do this, he first diluted the sample by a factor of 20 times, not 200, 20 this time. He then made the white blood cells clearly visible by using a stain that makes the nuclei appear dark blue. Yeah, that probably binds to the DNA, by the way. Red blood cells don't have a nucleus, so they wouldn't stain dark blue. When counting white blood cells, the doctor only diluted the blood sample by a factor of 20 instead of 200. Okay, so why is this? The answer is just that. There are far fewer white blood cells than red blood cells in your blood. I don't have the exact number, but I would suggest that it's probably at least 1,000 times lower. Yeah, they're probably around about 1,000 red blood cells for every 1 white blood cell kind of floating around. 4.5. Explain how the stain allowed the doctor to count the white blood cells amongst all the red blood cells. That's a strange one. I think this is just a mark for saying that red blood cells do not have a nucleus. Because the stain binds to the nucleus, red blood cells do not have a nucleus, therefore he could easily distinguish which ones were the white blood cells and which ones were the red blood cells. Yeah, let's just double check the mark scheme for those two bits. Except converse for red cells. Okay, cool. Yeah. So there are fewer white cells, so you need to dilute. So no need to dilute further. I'm not sure if I would have got this so. Maybe when I wrote it down I would have done. There are fewer white cells, so we don't need to dilute as much, or if we did dilute as much, we might not even have any white blood cells on the slide to see. And then for this one, white cells have a nucleus, or you could say that red blood cells do not have a nucleus. We say both, just to be sure. Okay, let's keep going on to question 5. Scientists investigated the role of a protein called SENPW in mitosis. Their method involved cell fractionation and ultracentrifugation. The scientists began by lysing cells and organelles using a detergent that dissolves lipids in water. Okay, so this is just saying, okay, the detergent dissolves lipids in water. So what lipids are we talking about? So the detergent dissolves the phospholipid bilayer that forms the outer membrane, so that, well, I probably and all this, also the organelles. It says cells and organelles. So it would dissolve the outer membrane and the membranes surrounding membrane-bound organelles, thus releasing the cell contents into solution. Okay, on to 5.2. I'm dropping in a little picture here to help us understand this process of ultracentrifugation. I'll just leave that up there. So explain how ultracentrifugation separates SENP from other molecules. So what we've got here is the homogenate. This is when the cell has been broken open. We've got a bunch of different organelles all mixed in. Ultracentrifugation separates things according to size or mass or density. So what we do is we spin at very high speeds in a little machine that whizzes the cell contents around, and the heaviest, largest, densest things sink to the bottom first. You do need to sort of know the order of these fragments. So the thing that would sink to the bottom first is the nucleus, and then we take the supernatant, which is the liquid part, and we put it in a new tube and spin again at higher speed. The thing that then comes next, if it was a plant cell, would be the chloroplast. And then the thing that comes next, in any cell, would be the mitochondria, and so on and so on. Finally, we're left. Well, then we get the ribosomes out, and if we did this the right number of times with increasing speed, we could eventually separate out just the liquid with dissolved proteins, and potentially even get SENP itself if it's a particularly large protein. Okay, so check mark scheme. Focus on exact keywords. Here we go. So spin at high speed is what you need to say. That is the definition of ultracentrifugation. Spin at high speed. And then the molecules separate according to either mass or size or density. On to the next question. Okay. SENP is involved in the formation of spindle fibers in mitosis. So remember, spindle fibers are the things that pull the chromosomes apart to opposite poles. And they're made of a protein called tubulin. Spindle fibers are actually kind of microtubules, which are made of tubulin, and they grow out from those centrioles. The scientists treated cells in a culture with small interfering RNA. This siRNA causes RNA interference of expression of the SENPW gene. Somebody was involved. Okay. The scientists took samples of cells at 0, 48, and 72 hours after adding the siRNA. The scientists took samples of cells at 0, 48, and 72 hours after adding the siRNA. They then used gel electrophoresis to separate SENPW and tubulin from 3 samples. Figure 5 shows the results of the electrophoresis. The size of each band is proportional to the amount of either SENP or tubulin present. Okay, so what I'm noticing is that the amount of SENP decreases pretty consistently to 72 hours. The amount of tubulin decreases pretty consistently to 72 hours, maybe even slightly even more reduction than there is SEN-P, just a little bit. This is a very thin line here. Suggest how the siRNA produce these results. So what we need to say is siRNA is an RNA that is complementary to the messenger RNA of the SEN-PW gene. So the two RNAs bind together. This is the mRNA, this is the siRNA, come together, producing double-stranded RNA that is then broken down in the cell, preventing the translation of the SEN-PW protein, and that prevents the spindle fibers forming. Let's check the mark scheme. Okay, so siRNA binds to the mRNA, prevents translation. This, I'm not sure if I would have got the mark the way I said it. SEN-P reduces, and so does tubulin production. Now this is interesting because in the text it says that SEN-P is involved in the formation of the spindle fibers. It doesn't actually say it's involved in the production of tubulin, but I guess you can assume that. Tubulin is the monomer, and the spindle fibers are like a big polymer of it, or an aggregate. So, but I can see what they're saying. So I guess we're maybe assuming that SEN-PW is a transcription factor. It doesn't say that, but it sounds a bit like that's what they're suggesting it is. Okay, so does reduce tubulin production. Question 6. Ammonia in soil is oxidized to nitrites and nitrates by species of nitrifying bacteria. So NH3, 2 nitrites is NO2 something minus, and NO3 something minus. Can't remember the charge actually right now. But ammonia, nitrites, nitrates. Nitrifying bacteria. Scientists investigated whether two soils with a different pH contain different communities of nitrifying bacteria. These communities consist of all the nitrifying bacteria of different species in each soil. Okay, so we're kind of looking at maybe biodiversity almost. They took samples of soil from 2 sites, A and B. They measured the pH of the samples and found that A had a pH of 6.9, so just below neutral, and B had a pH of 4.3, so acidic. The scientists measured the concentration of ammonia in soil samples over 20 days. Okay, so we know the duration of the test. So it's not very, very long, 20 days. Maybe that's going to come up later. Each sample contained the same concentration of ammonia at the start and had the same mass. Did they add this ammonia? Okay, I guess it looks like they maybe added the ammonia at a fixed concentration and then sort of could tell how quickly it was broken down. It looks like that's what they did. It doesn't say that. They recorded the concentration of ammonia in soil A and B, and this is weird. What's this? A mixture of equal masses of soils A and B with its pH adjusted to 6.9. So this is some form of control. It's a bit strange, but fine. Okay. Okay, concentration of ammonia. All right, so A goes down, B goes down faster, and A and B does not go down very well at all. Interesting. Okay. I'm looking at the lines for A and B, by the way. B is really rapid up until day, what's this? Is that day 2? Yeah, I think that's day 2. A, pretty rapid up until day 3, I think. Yeah, 1, 2, 3, 4. Yeah. And then it kind of slightly slows down up until day, I'm guessing that's 9. Yeah, that is. And interestingly, the slope of this one, the A plus B, is more or less this. It kind of more or less follows the same pattern, except just shallower gradient as A. I don't know if that's going to be relevant. I'm just kind of looking at the trends. Let's look at the question. The scientists use units of micrograms per gram for the concentration of ammonia in soil. So suggests why in this investigation the scientists use these units. Micrograms, well, this is just a 2 marker, so I think it's quick answers. So there are small amounts of ammonia in the soil. That's it. Microgram is a measure of a small amount. Small mass of ammonia. Maybe we should say it's small mass of ammonia in the soil. And this per gram, so we can quantify, it's, well, because we're looking at ammonia in soil, it's kind of like a concentration of ammonia, but we're not, the soil is not liquid, the soil is a solid, so it's looking at something per gram is a way of getting a concentration of the ammonia. I don't know what the mark scheme is going to say for the second bit, to be honest. I'm a bit lost. Okay. To allow comparisons. Anyway, so 6.1. Very little ammonia in soil. That's the micrograms. Or you could have said, okay, avoids use of lots of decimal places. If you're using grams of ammonia per gram of soil, it would be lots of decimal places. So this is a stock phrase to remember, to allow comparisons between samples. To allow comparisons. So maybe a standard form of question, why using this unit? Standard things to remember are to avoid lots of decimal places, or because there's little quantities, and to allow comparisons. 6.2. Calculate the difference in the rate of breakdown of ammonia per day between day 0 and day 2 in soil A versus soil B. Okay, fine. Difference in rate. We're basically looking at the gradient. Was it between day 0 and day 2? Yes, it was. Day 0 and day 2. Okay, so first of all, let's look at soil A. So we start at 20. So the change in Y is going to be 20 minus whatever we get to at day 2. Day 2, this is an annoying scale, isn't it? How many actual lines are there? I feel like my PDF is missing one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 lines up until this point, 20 lines up until this point. So that means it's 4 lines, 1, 2, 3, 4, per day. 1, 2, 3, 4. Okay, so this is definitely 2. Just checking that I'm measuring it at the right point. And we're looking at right there. And then we want to go across, and that's at 2 lines up. So that's 11, because that would be 12, 13, 14, 15. Yeah, so that is 11. So 20 minus 11. And then we're going to divide that by 2. So that is 9 divided by 2, which is going to equal to 4.5 micrograms per gram per day. That's the unit. What about B? Let's change color. B, we're going to again start at 20, and we're getting down to, where are we getting down to? At 2, we are at 1, we're at 2. We're at 2. So 20 minus 2 divided by 2, so that's 18 divided by 2, so that is 9. So it's double the rate, actually. So 9, and again, that unit is micrograms of ammonia per gram per day. So 4.5 versus 9. So the difference is 4.5. 4.5, get the unit right, micrograms per gram per day. Difference. Let's check the mark scheme. Yeah. Ooh. Okay. I think I'm going to get 2 marks for this, but to be honest with you, I don't know what the marker would say. Do you actually have to write out day to the -1, or would d to the -1 be allowed? I actually don't know. I guess write day, just in case. I'm not sure. I think it would be credited. I'm not entirely sure. Okay, 6.3. The scientists concluded that the soil mixture experiment showed there were different communities of bacteria in soils A and B. Oh, yeah, I think this is true. How can I express it? So I think it is that the community in soil A and the community in soil B are kind of each adapted to work at the pH of that soil. So you might think that if you had a mixture of soil A and B with a pH adjusted to 6.9, well, I suppose you'd only have 50% of the soil B bacteria in that mixture. And those ones are kind of working. Yeah, that makes sense. Okay, so here's what I'm suggesting. The gradient of this one, that's soil A, is about twice the gradient of this one. And that makes sense because in this one, the mixture, we're just taking, it's a 50-50 mix of soil. So we've only got 50% of the bacteria, of soil A bacteria in that mixture, and they are able to work as normal and break down the ammonia. Hence, the gradient is like half the gradient of soil A. But it would appear that all of the bacteria in soil B are basically not doing anything because the pH has been adjusted to 6.9, so those bacteria are actually not adding anything, they're not adding any extra breakdown. So that is how would I write that? I would write it something like that. Let's see what you get marks for. Okay, yeah, that's kind of what I was saying. So pH, so B has the fastest rate of breakdown. You get a mark for saying that. And A and B, a mixture at 6.9 has the slowest, is slower than A or B, and that suggests that that the bacteria from soil community B don't work well at the adjusted pH of 6.9, except the converse. So it's, oh yeah, okay, that's kind of what I was arguing, that only the bacteria from community A is working in the mixture because it's at the pH that it is used to, but it's kind of half the rate because it's only 50% of the bacteria. So that would be credited. Okay. What else? Okay, another part to this. The oxidation of ammonia by nitrifying bacteria involves the enzyme ammonia monooxygenase. Each species of nitrifying bacteria has its own specific AMO-A gene that codes for the production of ammonia monooxygenase. In a second investigation, the scientists determined the expression of the AMO-A gene in 2 species of bacteria, S and T. Species S was from acid soil, highlight that, and T was from a soil with neutral pH. Okay, the scientists grew cultures of each species separately in soils of different pH. They determined the amount of mRNA from the AMO-A gene in each culture. What on earth is this? Okay, so copies of the mRNA per gram of soil. Okay, species S from acid soil, as you would expect, produces lots of mRNA in acid soil, and it produces less and less mRNA as the soil pH increases. So that's going down. Right. And then the other one, the species from the alkaline soil, sorry, the neutral-ish soil, it's increasing as we're getting closer to neutral. And again, it's the mRNA that is increasing. Now, this is weird because I might have thought that if I was just guessing as to this question beforehand, I'd be thinking, okay, pH changes causes the enzyme to denature, but it seems to be something else. pH is actually affecting the transcription of the protein, not just its eventual functioning, whether it's denatured or not, if you see what I mean. So something is going on at the level of transcription. That's kind of strange. In which species was the number of copies of mRNA more affected by change in soil pH from 4.9 to 7.5? Use a calculation to support your answer. This means that we're just going to have to do maths, okay? We have to do maths to get marks. 4.9, 7.5. Okay, so we're just looking at the logarithms. That's why this question is there. Okay, so this one, the pink species, the acid soil, goes from 10 to the 6 to 10 to the 4. So that is a 10 to the 2 reduction. We can just use powers, easy. So that's 100 times reduction. What about this one? This one, the green one, goes from 10 to the 2 up to 10 to the 4. Oh, this is annoying. So that is again 10 to the 2. So that is 100 times increase. So which one is more affected? They're affected the same. This is silly. Let me just check the question again. .9, 7.5. Number of copies of mRNA. The number of copies. Well, okay. So it's in terms of multiplication, it's the same effect, but this is so silly. So this is basically going from 100 up to 10 to the 4 is 10,000, i.e. a change of 9,900. Okay, that's the change, delta. Whereas this one is going from 10 to the 6, that's a million, down to 10 to the 4, which is 10,000. So that is a drop of 990,000. So this has changed by 9,900, and this has changed by 990,000. So therefore, the answer is the acid soil. What a horrible question. So, species S has a change of 990,000, whereas species T has a change of 9,900. There we go. Accept standard form. If you got as far as I did when I got halfway through and then got a bit stumped, it says except for 1 mark, 100 times greater or 100 times smaller, incorrect context with no other calculations shown. And then I realized that I had to actually kind of talk about the numbers rather than just the multiplication, if you see what I mean. All right, let's keep going. 6.5. This method allowed the scientists to estimate the expression of the MO gene in each culture, not the growth of the bacterial population in each culture. Explain why. Okay. 4 marks, I guess 2 parts. Estimate the expression of the MO gene. So maybe there's some marks for that, and then some more marks for, but not the growth of the bacterial population. 2 marks for each, maybe? I don't know. Estimate the expression of the MOA gene. Well, because we can assume that the mRNA that is produced, call it mRNA? Yeah, the mRNA that is produced is going to be translated. So there should be a direct correlation or a linear relationship between the number of copies of the mRNA produced and then the amount of protein, AMO-A, or the ammonia monooxygenase protein produced. Okay, so there should be a direct relationship. So that's kind of tackling the yellow part, but not the growth of the bacterial population. Let's go back up and look at the method. They grew this is the key bit here. They grew cultures of each species separately in soils of different pH. They determined the amount of mRNA. It doesn't actually say how. But we weren't looking at there was no measure of the size of the bacterial population. There was no counting of cells. There were no counting of colonies. Yeah. And it was only done for 15, was it done? No, it was only done for 20 days. I'm stumped. I'm going to look at the mark scheme. Okay, yeah, I mean, that's what I said. I didn't think that was going to be even worth saying, but always say the obvious. It can get you marks. They didn't count the bacteria. Therefore, they didn't know the size of the population. And the number of mRNA should be related to the amount of the protein translated. This is some good points here. I was thinking this, I just didn't know if it was going to come up. So when you're growing the bacteria, what's changing? Let's look at this graph one more time. This is the copies of mRNA per gram of soil, which is weird. So is it that the bacteria are growing and the rate of production in every single cell is just staying the same? So like if we're looking at this green one trending here, we're just getting more division of cells as the pH of the soil increases, but the frequency of copies of mRNA in each cell is the same? Or is it the opposite? Is the rate of growth of bacteria staying constant, but the expression of mRNA is changing? And we don't know. That's the answer. We don't know which one it is. Seems like a strange method. Yeah, this is a sensible thing to say. We don't know how much mRNA is being produced by each cell, and we don't know. Strange one. We don't know if AMO-A is linked to cell division or growth of the population. I don't quite follow that one, to be honest. But I can understand we don't want to talk about mitosis and meiosis because this is binary fission. These are bacteria dividing. We're not dividing a nucleus. I don't know what they mean by 4, to be honest. Okay, 6.6. The scientists set up their cultures in sterile glass bottles. Suggest one suitable method for sterilizing the bottles and explain why it was necessary to sterilize them. Okay, so you could sterilize them in an autoclave. Would that give you marks? I hope it would. So an autoclave is basically like a kind of like a steam bath, but very hot steam at above 100 degrees Celsius. Autoclave the bottles so that all the bacteria on the bottles are killed. And why? Explain why. This is to prevent contamination so that no other bacteria grew in the culture, and it was only the specific nitrifying bacteria that they wanted to look at were cultured. Other than autoclave, if you didn't remember that, I'm imagining flame sterilization would be good. Okay, autoclave, yeah. Autoclave. Alcohol also would be good. It does say ignore flaming of the bottle. Okay, I can actually understand why. Because if you're flaming the bottle, that is a procedure that you do to sterilize the ring of the bottle, the lid of the bottle, as you remove it. So quite often you kind of take it off like this with one hand and you remove it, and then you flame the lid. So you flame the neck of the bottle, and then you put your lid back on and you seal it. But it doesn't actually sterilize the inside of the bottle. To sterilize the inside of the bottle, you need to take it apart, take the lid off, put the whole thing in an autoclave, run it, and that will kill other bacteria. So, now, okay, so you have to have this. You have to have this second part here for this mark. So, yeah. So ignore contamination unqualified. You've really got to explain why it would be of significance to the results I'm learning. So you can't just say to kill other bacteria that might contaminate the culture. You have to say why that would be a problem. And it's because if you're trying to quantify the rate of breakdown of ammonia, you don't know what the other bacteria are going to do. Are they also going to break down ammonia? They might even make ammonia. You have to really articulate how it would affect the results. So you don't get this, ignore reference to bacteria that would affect the result. You have to say how. So you could say that these other bacteria might break down ammonia. You could say that they might compete with the nitrifying bacteria, or they might even produce ammonia. So there's kind of three options that these other bacteria how they might mess up the results. You've got to say that, something I'm learning. Right, now we get to the essay. So I actually advise you, if you're doing this paper for real, my advice is to turn to the essay straight away as soon as you open the paper and have a look at the two essay titles. So in this case, the importance of the control of movement in cells and organisms, and the importance of interactions between cells and between organisms. And to start with, you should spend about 3 minutes doing an outline plan for each one. And then you can select which essay you'd want to do. So let's do that together. Here's how I would be doing this. Let's go for control of movement in cells and organisms. Okay, so first of all, movement of organisms. Muscles is an obvious one. And control of movement. So nervous system, action potential, and then muscle contraction. That's 2 areas. Movement within cells, maybe we could think about mitosis or meiosis, because that is movement within a cell. That's movement of chromosomes within cells. What else? Movement within cells, we could also have stuff to do with cytokinesis. Sorry, not cytokinesis. Phagocytosis is probably what I would go with. So the white blood cell being able to move towards a pathogen, to engulf it, to digest it, that's all movement. Other things that are now coming to me, movement of cells. We could talk about, ciliated epithelia in the respiratory tract. That's now giving me my 4 areas. And you should probably also think about how you can sell the importance of each thing. So why are each of these things important? The nervous system, the importance of sending an electrical impulse to the right spot at the right time so that only one muscle is moved to generate the right response. Muscle contraction, the importance of being able to control your limbs through antagonistic muscle pairs, being able to contract one muscle whilst allowing another to relax, to allow the coordination of the body to generate the appropriate response. You could link this to maybe taxis and kinesis responses and stuff like that. Ciliated epithelial cells, the importance of removing pathogens from the lungs, removing dust from the lungs, providing a clean gas exchange surface that is not damaged to provide enough oxygen for respiration, and so on and so on. Oops. And mitosis, to precisely control the movement of chromosomes so that each cell gets an exact copy of the DNA. Phagocytosis, to allow white blood cells to engulf, digest pathogens, and then also to present the antigens. So I think there's definitely stuff we could say there. That's a doable essay. let's move on to the next one. The importance of interactions between cells and between organisms. Between cells and between organisms. So let's go between cells first. Okay, the immune system. There are so many interactions in the immune system. We're talking about T helper cells, B cells, T killer cells, antigen presentation. There's loads of cell-cell interactions there. Also, again, nervous system I would probably go for, because we're talking about how synapses allow communication between cells. Summation, one-way flow of information. I'm kind of talking about the importance here. Also, importance of immune system. Again, we're talking about guiding the immune system to either go for a cell-mediated response, that's T killer cells, or humoral response, that's B cells and antibodies. So that's kind of an importance angle. Doing the right response for the right pathogen, keeping you alive. There's also loads of stuff about between organisms. We can talk about food chains and energy flow. That's an interaction. Eating. We could talk about nitrogen cycle if you wanted to. Interactions between the organisms and the nitrogen cycle. We could also talk about kind of ecosystems in general and how biodiversity affects the stability of an ecosystem. So again, and then that has crucial importance for the kind of the global ecosystem, the world, being a stable place to live for everyone, humans and animals included. So once you've done that outline plan, did that take me 3 minutes? It might have taken me a bit more because I was kind of talking about it. Once you've done that outline plan, remember I'm suggesting that you do this right at the beginning of paper 3. Then I would say, just forget about it. Go and do the whole rest of the paper. So you would turn and go through the paper. Because as you're going through the paper, you might think of things. For example, when you're doing this question, you're thinking, oh, antibodies. Okay, antibodies, interactions between B cells and antigen-presenting cells. That's something I could talk about. And then when you get down here and into the nitrogen cycle question, you're thinking about the interactions between organisms of the nitrogen cycle. So even if you were slightly struggling at the beginning, when you do the paper and then you get back to your outline plan, sorry, it's taking ages, then you've kind of already done some thinking subconsciously whilst you're actually doing the essay. So then you're going to select the essay. And remember, when you select the essay, you must have 4 key areas that you can talk about with clarity, in detail, using the right keywords. That's kind of all the AO1 part. But then you also need to think, do I have a clear importance angle for each paragraph? The easiest way to do that is kind of like a domino effect type thing. So, for example, let's talk about which one? Let's talk about ciliated epithelia. How would I sell the importance of this angle? So the easiest way of doing this, I like to call it a domino effect, is think about what would happen if ciliated epithelia stopped or didn't exist? What would be the consequence of that? And then what would be the knock-on consequence of that? And then what would be the knock-on consequence of that? So ciliated epithelia, they are important in trapping dust and pathogens. If they didn't trap dust and pathogens, then dust and pathogens would land on the respiratory surface. They'd make it into the alveoli. This could cause more frequent infections of the lungs. This could cause scarring of the alveoli, such that the person had a reduced lung capacity because of damage. Reduced lung capacity would reduce the surface area of the gas exchange system, which would mean that less oxygen could be absorbed with every breath, which would mean there would be less oxygen to act as the final electron acceptor in aerobic respiration. So that would mean less ATP, less energy available for metabolic reactions and for life. Okay, so that's like a small thing, how it can have a big consequence, but you have to follow the kind of chain of events. Okay, I'm going to give you one example paragraph that I'm going to write about a different area, and I'm going to pop it up and then you can read it. Okay, I'm actually halfway through the paragraph that I'm writing, and I wanted to pause and stop because I've gone down. I've done something bad. I need to stop myself, and this is so easy for you to do. I've started putting too much in. I want to talk about the interaction between cells, and I thought, oh yeah, I'll talk about phagocytosis first. And I've suddenly realized that I'm putting too much detail in, and this is like a big thing that you need to learn to avoid. You want to get straight to the most critical bit of the AO1 that you want to describe. You don't want to just say, okay, there is some interaction in the immune system, so I'm going to describe the whole immune system. It's too much. So I'm actually going to probably start again. You can pause and read what I wrote if you want, but I'm going to start again, and I'll show you my second draft in a second. Okay, so here is my second draft of my paragraph talking about how the immune system, or the importance of cell-to-cell interactions within the immune system. Okay, so I'm going to pause, or you should pause, read this, and see if you can sort of figure out where I'm using keywords, where I'm kind of emphasizing my AO1, my content knowledge, and where I'm emphasizing the AO2, the importance of this. Once you've done that, we'll annotate together. Okay, have a go. Okay, so here's my kind of self-annotated paragraph, just to show you kind of how I was trying to emphasize different assessment objectives within it. So yellow for me is AO1. Okay, so I've got AO1 here demonstrating my content knowledge, and I've also highlighted in bold here the keywords that I'm using. So I think to really sell your knowledge of AO1, you need to be using A-level vocabulary. And a big mistake is students to kind of write in more general terms, maybe even kind of dipping back into kind of just GC level of vocabulary, and this isn't credited. So you need to make sure that you're using the A-level vocabulary, the keywords that you have learned. So that's my AO1 section there. I then move into some AO2 down here. This is about the importance of the what would happen if cell interactions weren't part of the immune response. I kind of then go back into a little bit of AO1. This kind of maybe a bit of AO1 / AO2 actually, really, this little bit here. Because I'd kind of forgotten to talk about chemical signals and how they regulate cell-to-cell interactions. And then I'm kind of having to make a bit of a hypothesis almost about what would happen if there weren't cell-to-cell interactions in the immune system. What if we only had the same immune response for every single pathogen? What would that be like? But you can see the balance of AO1 and AO2 in this paragraph is roughly 50-50, and that makes it an effective paragraph. I probably spent a little bit too long on it, but if you can kind of produce 4 paragraphs a bit like this, then you are going to get in that top band of marking. Okay, so here is the marking guidance for the essay. We can see that there are a few different bands. Nationally, the average band for people to get in the essay is just this multi-structural band. And you need to remember that to get in here, you need 4 different topics, 4 different paragraphs from different areas of the spec, even to get to this point. If you've only got 3, then you're going to be capped at unistructural. So how do markers try and place you in a band? So 4 good relevant paragraphs with strong AO1, but your AO2 maybe is not brilliant, a bit waffly, that's going to land you in multistructural. If you want to get in the next band up, you're going to have strong AO1 and clear AO2. Maybe if you're at the low end of this band, then 1 paragraph is letting you down a lot. Maybe it's kind of irrelevant, or there's some significant errors. At the top of this band is great, 4 paragraphs with great AO1 and clear AO2 for all of your paragraphs. Up here, this band, where everyone wants to be, it's very hard to get in this band. You need excellent paragraphs. I would say 4 excellent paragraphs. There isn't actually no need for 5. You need hardly any irrelevant sentences. You really need to be concise in your writing and just get to the relevant points, not just describe everything that you know. I almost made the mistake of this when I was doing my paragraph. I started to talk about phagocytosis and I thought, hang on a minute, this isn't really relevant. And then I kind of actually decided to leave that out. So maybe if I'd put too much on phagocytosis, I would have brought myself down a bit. So you want to be concise, you want to be targeted. Right at the top, you need 0 waffle, very precise, and you need a little bit of post-spec stuff. In the paragraph that I'll show you again in a minute, there's a tiny bit of post-spec I put in about MHC class 2 proteins, and I think interleukins and cytokines is a bit beyond spec for AQA. It is on OCR. So maybe that might have got me up there, but I need some more. Down at the bottom, I'll let you read that. And a good thing to think about if you've got an essay and you've written it and you want to kind of evaluate it is, what do you think, reflecting back on it, what do you think is your best paragraph? What do you think is your worst paragraph? Why is your best paragraph better than your worst? And then how could you improve your paragraphs? So that concludes our run through of this paper 3 from 2018. I hope you find that helpful, and best of luck with the revision.